So what I wanted to do today was to actually give sort of a general introduction into gene therapy. I know the audience is sort of a mixed scientific background, but rather than going into the very fine details of what my research lab does, I wanted to give sort of a general overview. I will, sprinkle into the final sets of the slides some of the work that we've recently worked on. But the other thing that I wanted to do was, again, to highlight, how PacBio long-read sequencing has enriched this field. So just a general, again, an introduction of gene therapy, and I'll need to put on my glasses so I can read. But gene therapy seeks to modify or manipulate the expression of genes or to alter the biological properties of living cells in therapeutic use. And I think this is a perfect, sort of placement for my talk following Lysenka's introduction into rare diseases, because these platforms really do excel at treating monogenic diseases, which fall into the class of rare diseases. So they can act to replace genes or faulty genes that cause disease, or they can be used to silence genes that have mutated to then become toxic to cells, or you can add exogenous genes in order to change the profile of the tissue for therapeutic purposes. You can also edit genes, right? So I like to classify this as gene addition, but because these lead to permanent manipulations of the gene in the patient's genome, they really fall into the class of their own. So gene therapy really is, at the heart of it, delivery of its payload. And typically, these are done through vectors. And the most promising vectors, as it stands right now, are those that are based on viral vectors. They're typically removed of any genes that make viruses pathogenic, and in their place they are engineered with therapeutic gene products. So there are multiple viral vectors that are on the market and being researched in preclinical and clinical trials. Illustrated here at the top. Let's see, yeah, it's barely visible, but as you can see here, even in this summary in 2021, the predominant viral vector that's being used is based on AAV, the primary vector that our research group works on. And then there are also adenoviral vectors, which are large DNA viruses, herpes simplex viruses, and then lentiviruses, some of these vector platforms you guys might have heard of before. And most of these are in phase 1 and phase 2 clinical trials. And for the disease targets that they tend to be developed towards is really diseases of the eye, diseases of the brain or the CNS. There are gene therapies that are also being developed for metabolic diseases, as well as those that target blood disorders. And then, of course, musculoskeletal diseases. So, as I mentioned before, AAV, or adeno-associated viruses, have become really the leading platform for developing gene therapy drugs. And so I was just going to sort of give a general introduction for why this is so. They belong to the dependoparvovirus family of single-stranded DNA viruses. So on their own, they're non-infective. They actually need what's called a helper virus. They were actually first discovered as a contaminant in the 1960s by Bob Acheson in adenoviral preps. So they're very, very small. So actually all of these bigger particles are adenovirus particles. The black arrow in the top part of the graph is actually the AAV particles. So these are very small, 26 nanometer sized viruses. They're considered to be non-pathogenic and have low immunogenicity. And as I had mentioned before, they're non-replicating on their own. And they're known to be benign. About 40 to 80% of the human population, depending on the community, they're already seropositive for AAV. So they're known for these specific bullet points to be the ideal gene transfer vector. So the anatomy of a typical AAV vector is pretty simple. One of the limitations, of course, because of the small particle, the vector cassette has to be under 4.7 kb, which is the size of the wild-type AAV. And the only elements that are retained from the virus itself are these T-shaped hairpin sequences that are flanking the genome. They serve as origins of replication, as well as the packaging signal for producing the virus. And as I'll mention near the end of the talk, they're actually required for stabilization of the genome so that these therapies are considered as one-shot therapies for the rest of the patient's lifetime. So the cassettes are fairly simple, right? This is just one sort of typical design for a protein coding gene. You can have the therapeutic transgene, a promoter that drives its expression, and any auxiliary elements that allow for its robust expression profile. Currently, there are 7 gene therapies that have been approved commercially. The first was Luxturna for treating Leber's congenital amaurosis, which gained FDA approval in 2017. And then 2 years later, Zolgensma, which is a gene therapy for spinal muscular atrophy, was approved in 2019. In 2022, we saw approval of Hemgenix and Roctavian, which are gene therapies for hemophilia A and hemophilia B, respectively. And then in the EU, Upstaza was approved, which is a gene therapy for AADC deficiency. Last year, we saw an approval for Alividis, which is a gene therapy for Duchenne muscular dystrophy. And then the beginning of this year, there was a second gene therapy for hemophilia B, Becfez, that was approved in Canada as well as here in the United States. So there are more and more of these gene therapies that are being tested. And so among the 4,000 therapies that are in preclinical through pre-registration, about half of these are fallen into a category of gene therapies and gene and cell therapies. So, there is definitely a lot of promise for these type of platforms, and a lot has been happening in the past few years for these magnificent therapeutic strategies. So in 2024, there was a general increase in the number of gene therapies at each pipeline stage of development. So how have we been using PacBio long-read sequencing to sort of enrich these platforms? So in the past few years, we've been developing and trying to standardize vector QC methodologies. Because one of the things that we now realize is that when you produce viruses or viral vectors, they're not as homogeneous as anticipated, right? We all know that viruses can have mutations and they generate defective interfering particles because a super good virus is going to kill the host. And so viruses have come up with methods in order to generate a bunch of defective particles. And I think this has sort of been a carryover of these sort of mechanisms. So although we would like to have drugs that are homogeneous, they just tend not to be. And this has really changed how we consider safety profiles for these vectors, and still this is ongoing work from our research lab as well as other labs in the field. We've also been using PacBio sequencing in order to gauge potency. For example, gene therapies that apply gene editing, we'll use PacBio in order to profile, long deletions as well as indel events, and that's been very, very useful in this area. But one of the things that I wanted to focus the talk on now is really new biology that is learned from PacBio sequencing, and that is ITRs, right? So I get this question from people that are not in the AAV field multiple times, right? So again, ITRs are those elements that I'd shown that was flanking the vector genome that remains the last element that comes from viruses, right? The reason why they are important for me, at least, is because these elements, due to their strong secondary structure, they cannot be sequenced by any other technology aside from PacBio sequencing. And when we first approached different sequencing companies about sequencing through ITRs, they always just told us, well, just cut them off, right? But as you'll see, these things are super critical, and it's really driven some of the science that is happening in the lab. And so if you guys have ever sequenced AAV, you'll find that a lot of the ITRs are mutated, right? So is this necessarily a good thing or a bad thing? So just a little bit of background about ITRs, right? So again, they're these T-shaped hairpins. I don't know whether you can see from the back there, but they have what's called an A region, which makes up the stem of the T shape. And then on the arms, there's a B arm and a C arm, which are sequence unique. And then on the far end, there are the single-strand D elements, okay? And ITRs exist on, again, both sides of the genome. And between the A sequence and the D element, there is a terminal resolution sequence. So this sequence allows for replication proteins to nick, and that continues the DNA replication cycle, which I'll discuss. There are generally two configurations for AAV called flip and flop. Flop is where the C arm is closest to the open end of the genome, and then flip is where the B arm is closest to the genome. And all parvoviruses have ITRs and they have different configurations, but AAV's sort of unique that it packages both plus and minus strand genomes at equal ratios, and the orientations, where the configurations are actually independent on each end. So you'll have flip, flip, flop, flop, flip, flop, flop, flip at 1 to 1 to 1 to 1 ratios for a genome that's properly replicating. So I have the slide started to illustrate, but if it's hard to follow along, I can talk to you about this afterwards, but it's really interesting. So basically, when you design AAVs on plasmids, right, you can have single orientation ITRs. So for example, on a plasmid, if you rescue a vector that has flip-flip, actually after synthesis of the second strand, you'll get this double-strand molecule illustrated on the bottom here. Yeah, laser's not working. But then on the 3' end, after you get nicking, that 3' ITR then becomes a new 5' end, and that's where you have this configuration be flipped. And so at the end of this first replication cycle, you'll have a positive strand flip-flop oriented molecule, and then a negative strand flop-flip oriented molecule. And this can proceed multiple times. And so after about 4 generations of replication, you'll have covered all 8 configurations. And actually, when you look at your genomes after PacBio sequencing, you can actually gauge how well the vector genomes have replicated. And we actually use this sort of as a dirty way of looking at how well AAVs have been produced and packaged. And so it's all fun to know this sort of information, but how does this really impact the vectors that are packaged? Well, it turns out that they do lead to a degree of heterogeneity that's packaged for AAV. And some of this work came from originally from a collaboration that we had with Magali Budlu and Edward Aoso a few years ago. And they were interested in looking at heterogeneity that's defined by different packaging mechanisms. So there's 2 main ways that AAV is now being packaged for clinical vectors. One is to use standard transient plasmid transfections in human cells. That's illustrated here at the top of this cover that we had in human gene therapy. And then the second is baculovirus infection of insect cells. And so what they were able to show was that using a genome that was similar between 2 packaging platforms, that the human cells, that vectors from the human cells were more homogeneous in terms of sequence length than those that were packaged in insect cells, which turned out to have a high degree of truncated forms. And I won't go into all of the individual figures that we came to this conclusion, but I will say that one of the reasons why we found where the major differences was the fact that in human cell produced vectors, the ITR remain these 145 nucleotide wild type length ITRs, whereas ITRs that were produced, or ITRs that were associated with vectors that were produced in insect cells had a high degree of heterogeneity. They were unresolved, they had these trident-shaped ITRs that were missing the B arm and C arm. And when we start to segregate some of these ITR classes, we find that those that are associated with mutations led to a high degree of unresolved genomes and a high degree of truncated genomes. And so how does this really impact sort of what the field is currently striving for? Well, because again, the ITRs are the last viral elements, there's always some goals to sort of engineer these ITRs to make them better and to enhance production. But if I said that in those previous studies and those of others that if you have mutated ITRs, you compromise packaging, how do we sort of deal with this and how do we analyze? And so, of course, we've also been using PacBio sequencing in order to gauge these. And it turns out that there are actually in the field some plasmids that have been circulated that have mutated ITRs. And so a lot of regulatory agencies have asked us, well, what does this mean for safety, right? And it turns out that for the most part, mutated ITRs are repaired. So this is something that is really great for people developing these constructs based on these plasmids. And this repair is actually dependent on the other ITR copy being wild type. So instead of undergoing standard rolling hairpin replication, which is the figures that I showed before, they actually use the opposing strands ITR to replicate and reform the wild type ITR. However, this isn't as straightforward, so recently we formed a collaboration with Carmen Unzo and her graduate student, Carla Ilnares, and what they wanted to know was whether there were certain factors that contributed to this repair mechanism. Because it turns out that different promoters that are being engineered into these vectors, as well as sequence length, actually make an impact on the rate of ITR repair. So the mechanisms driving these sort of different rates is currently unknown, and we're currently pursuing these questions. The other thing is that if you mutate both of the ITRs so that you don't have a chance of repairing, to restore the wild type ITR, that actually the repair gains the ITRs of the most functional ones. So even though you can have mutations on both ITRs, it's not like they're retained within the final vector that is produced. It actually will repair it to the most wild type or the most functional one that's within the cassette design. And so, yeah, I usually have this slide in the beginning of my slide deck because it really does illustrate sort of what the expectations are and what we actually see when we start to deploy these third-generation sequencing platforms. And one of the things that were gained from these types of investigation was that all of these sort of contaminants that we see in our preps are actually due to a high degree of recombination that will recombine, even DNA sequences that originate from the plasmids that are used, as well as genomic sequences coming from the host packaging cell. And typically, these are deemed as low risk, right? Because they don't carry full open reading frames. They just carry sort of small fragments. And it's been known for years that these things do persist in preps. But one of the things that we had revealed was that they're not packaged by passive packaging of DNA or elements that look like ITRs, because again, ITRs carry the packaging signal, but actually they're being recombined with ITRs themselves. So one of the potential thoughts is, how are these genomes persistent in the cell once they get in, right? Because again, the ITRs are critical for its stability, and yeah, I'll show that in the next, not this slide, but the following few slides. But the reason why I bring this up is because there's one aspect that I think not a lot of people outside of the field think about is the concept of replication-competent viruses. And so when you're producing viruses, of course, when you're producing these AAV vectors, you have all the components that are critical for packaging, right? You have the replication proteins as well as the cap open reading frame, which generates the capsid proteins. But it's been well known and well documented that at very, very low levels that you can get recombinations so that these components then restore the full vector, the full virus genome. And these can then take over the prep and then can potentially be bad as adventitious viruses which can cause potentially strong immunological responses in the patient. And so we started to sequence replication-competent AAVs because really the only way that they were originally detectable was to be passed through cell lines in the presence of a helper virus such as adenovirus, and then detect them by PCR. And a lot of entities, when they started to detect these things, they would make arguments, well, they may be contaminants that come from plasmid that are carried over, but really using long-read sequencing, we were able to verify that you do indeed capture these recombination events to restore AAV genomes. But one of the surprising things that we saw was that in a single prep, you can have a high degree of recombination events, suggesting that these recombination events are always happening. And it's just that, a right sort of combination of these events will restore the genomes. And so this suggested to us that there aren't any single motifs that drive recombination, and that this is really stochastic and can happen in any prep. And so I believe that these type of technologies are a means of being able to identify and verify that preps that are going to be used in patients lack replication-competent AAV. And so finally, this is the last area of work that I wanted to focus on. Actually, my graduate student, Suk Nam-Kung, received a PacBio Star Award to work on this. And this slide actually came from a review that we had published. And it's now, and it sort of describes a dogma of what genomes look like when they enter the cell. So again, AAVs for single-strand AAV constructs, once they get into the nucleus, they undergo second-strand synthesis to give you the double-strand DNA, or they undergo strand annealing. And so after this process, they can then become circularized genomes through the recombination of the ITRs to form stable episomal forms. They can also, again, undergo second rounds of recombination to form dimers and concatenamers. And they can also integrate into the genome at very low rates. And so these mechanisms hadn't been very well described because it's very hard to capture these type of genomes because of their low abundance. And in addition, without long-read sequencing technologies like one we have for PacBio, these sort of things were only detectable through methods such as southern blots, which hardly anyone does anymore, right? But what we found was actually quite surprising, because when we isolated livers from mice that were treated with AAV vectors and then subjected to rolling circle amplification and then sequenced on sequel 2, we found that regions that span the ITRs were completely missing. So this is really a paradigm-shifting finding, because one of the things that I didn't mention about ITRs is that they have been recently shown to have promoter-like activities. They're able to recruit transcription factors to drive and regulate transgene expression for vectors, as well as recruit epigenetic modifiers, right? So there have been many papers that have been published in the past and recently, but what this work is showing is that they're not there, right? So where does this activity come from? What does it mean for AAV biology? And we also know that the ITRs are also the elements that drive integration. So how does this affect integration, right? So I think using these sort of technologies has definitely helped us understand AAV biology. And with that, I'll wrap up. I wanted to really thank folks at PacBio. So many years ago, we had started this journey using PacBio sequencing. Michael Wayan, he actually came to our lab to build our first libraries. Cheryl Heiner, which I learned is recently retired. She was instrumental in getting this work started. Christina Weber, who was a bioinformatician, who started some of this work and helped us sort of understand how to do these, how to develop these workflows. And Matt Seaton, who also acted in that same sort of position to help us. I'd like to thank different members of my lab, as well as the GAL lab, and the Viral Vector Core for making all the vectors that was utilized in this study, as well as the University of Dinant, Magli Budlu's group, right, who helped us with the SF9 work. And that I'll take questions.